this going. Hi, people at home. If you wouldn't mind, if you're at home, just letting me know in the chat, here, present, whatever, something so that I know you're there. We always do it twice a day. Once right around now, right after the national anthem, 8:21 to whatever. And then we do it again after the break, which would be 11. So it's 11:05, but we go for a walk. Have you guys been going for a walk?
No.
No, okay. Do you want to?
Yes.
Okay, you do want to, okay, all right. So after we'll go for a walk. So like 11:15 or something, we'll come back. And that's, so I have to take attendance twice a day. So for you guys at home, that's like your main opportunity to let me know that you're following along with the class. But I do a second check right before 2:05, somewhere during learning block 3, just to see if you've been on Brightspace, and I count that too, but it's better this way because we had problems last quad with it not registering that people have logged in and stuff like that. So it's like that's not the best way for me to take attendance. So, but it's really the only other option I have. So anyway, just make sure you're here to let me know at some point that you're present. And you guys have it easy. You just have to physically be here. So no problem. I'm gonna do a little bit of housekeeping stuff here for the first little bit. I'm going to be here now for the rest of the quad, so we better figure out what we're up to. Did you guys all get textbooks?
Yeah.
You did? Every person has one? Okay. Did you sign up for your textbook online?
Yeah.
Everybody did it? Okay, so if you haven't done it, on the day 1 stuff here, there is where is it here? Day 1, go to content from the live stream. There is a section right here that says textbook sign up. If you haven't done that yet, you gotta go through and do that. I gotta get a registration from every person, so. But everybody has a textbook. Okay, that's the key thing. Because I don't have digital copies of all the textbook pages right to the end of the course, so I only have it for the first little bit. They were nice enough to give me those, because we mentioned it was hard to distribute at the beginning, so they said, okay, well, here's the first 2 chapters or whatever. Okay. Now for attendance. Okay, I gotta figure out who you guys are and where you are. All right, let's do this. Make a little seating plan. I did have a seating plan for you, but we'll have to, you guys at home have a seating plan because you were here on the first day, but so you guys are cohort B. Let's just see who's here for the ACRA. I don't know if I mentioned this previously, but if at any point you want to work from home, you don't want to work from the school, for whatever reason, doesn't really matter. You just have to make sure that your parents call in and let us know that you're working from home. It really doesn't matter. So the reason's not important, but they just want to make sure that you're not sick. If you are sick, that's fine too. You could be sick, obviously. I probably get sick too. Everybody gets sick in the winter at some point, but they want to track who's sick. So they want to know who's sick at which point, just to keep track of that. So just make sure that you're letting them know if you're not going to be here. I'm hoping in the second block that we're going to have more opportunity to do like group work, like small group work in here. I didn't do it last quad. I had no time to set it up, but I have a little bit more time this quad to like set stuff like that up. Do people in here have headphones? You do? Okay. So as long as you've got headphones with you, it should be okay. I've never, has anybody in their class ever done like people that are video conferencing in the room where you're in the room?
A little bit, yeah.
A little bit. How did it work?
Okay. Like we didn't do video conference, but we did calls.
Calls.
Like a few people from different groups, yeah.
All right. I'll give that a shot. I was a little bit curious whether there's enough bandwidth in here for everybody to do it simultaneously. That might be a problem. We'll see. I'm gonna.
Online sign up code.
What's that? Oh, it's like 2 pages in. One more. One more? No. Does your textbook not have one? Strange. Can you go back a page? There's like a, oh, it should be right here. Is it? Is it under that? Your textbook does not have one. Okay, I tell you what, you can use my online sign-up code. I don't use it. As long as you have the code, you should sign up for this too, just so you can access the online textbook. This is actually the code right here. It's Nelson8984GYDK. Use mine. That's weird. I didn't know we had a textbook like that. I better get that figured out. That means they stiffed us. We paid for that one, but they didn't give it to us. Yes. Yeah, of course.
Do you my name for attendance, or have you done that already?
I will need it.
Okay.
I'll get you when you come back. It's not a huge deal. Real quick though, did you fill out the little form online?
Yeah.
So that just goes for everybody in here. There's a little bathroom break form. You fill it. You guys already know how to do this. And then you don't have to ask. So you just go. Just go. Yeah. As long as you filled it out, I don't care. No problem. I trust you. Sorry, this is gonna take me a while. I don't know where everybody is sitting in my mental seating plan here. Okay, so what am I saying? Evan, LA, Nolan McNeil, Ross, Lou. Okay, now let's figure out the people that are here. You are?
Caleb.
Caleb. Apologies in advance if I know you and I just don't recognize you because of your mask on. I've been having a terrible time with mask face. Caleb, right? You said? Yeah. Cool. Okay, well, there's one. And Maria. Maria M, I'm assuming. Cool.
Katya?
Oh, Katya, yeah. It's the mask.
Jordan.
Oh, okay. I might figure it out mid-sentence too. Jordan and Julia, welcome back. Megan. Emily. Hi, Emily. Welcome back. I taught a lot of you guys in grade 10 probably, or grade 9. I don't remember anymore. Was it grade 10? Grade 9. Oh my goodness, time flies. It's Alicia. And
Andy.
oh, Andy, sorry. I feel like did you sit there last time, Andy?
I did.
And then Nolan.
Nolan.
And Carson's beside me.
Okay, cool. Just so you guys are aware, I'm sure you already know this, but once you've picked a seat, you guys are kind of locked in there, right? This is like your little germ zone, wherever you're sitting. Only your germs are in your germ zone. I do disinfect your desks every day at the end of the day, so they are clean when you come in. But this is like your little zone, so you can kind of feel safe with your desk and your chair. No one else will be touching it. So, and I do the chairs in between cohorts, so you can feel safe touching your chair and everything too. I really clean them well. So, sorry.
Oh, are you done?
Either way, it's fine.
Oh, Victoria.
Victoria. I think I may have had some of you guys in MSIP too. My memory's not as good for that.
It's Eva.
Eva. Miles.
Carly.
Carly. And Claire is not here? No. Did Claire sit there by any chance? Has Claire been here at all? Anyone seen? No one knows who that is? Claire McMillan? No? Okay. Just curious. Cool. Nice to see all of you in person. This real quick. I get much faster at this, by the way. Okay. Okay, we got that taken care of. Okay, next. Do you guys have any questions from, I guess, anything we did last week? Normally I would say from yesterday, but guys, was it okay? I know obviously it's not the ideal way to do that, but not terrible. I just noticed yesterday as I was going through everything that I forgot to edit out the part where I talked about the outbreak that we had last quad. Sorry if I freaked anybody out with that. That was from the previous quad. Other than that, it's all mostly the same. Okay, cool. Just a quick heads up to people, if you've, I think almost everybody's written the quiz from the first unit. If you have any problems with the quiz questions, make sure that you write down the question itself, not question 5. Because it randomizes the questions. So if you send me an email and say, oh, I didn't understand question 5, or question 5 has a weird answer or something, I don't know what that is. That could literally be any question. So just write down what the question is. I do make mistakes. There have been mistakes in them before, so it's not perfect. So sometimes I have to fix things. I gave some bonus marks last time because I found some mistakes in the quizzes. So if that happens, so don't be afraid to let me know, guys. You can definitely let me know. All right.
Congratulations for your baby.
Oh, thank you. Thanks. I do.
Okay.
Thank you. Do you guys already get report cards? Oh. Well, Brady got an early one. I don't know why.
Maybe this is from a different quad, I don't know.
It is. Oh. Your timing, I don't know. I guess some of them are early, I guess. Okay, so I mentioned the quizzes. The assignments are both, both assignments are due today. Yeah, by midnight. So it's usually the unit ends, and then there's a day, grace day, I don't know, whatever you want to think of it as, and then they're usually due at midnight at the end of the next day. So that would be the vocabulary list from the first unit. That's pretty straightforward. I mean, it's not the most complicated assignment you've ever done, I'm assuming. The key there is they just say you're doing things in your own words, okay? That's what I'm looking for. So, and that's how you're consolidating the knowledge, about that term or whatever from the first unit. So that's due at the end of the day. Usually people are just done that at the end of the unit anyway, because you just kind of just do it as you go. But just so you know, so that's at midnight. And then the other thing was the species spotlight assignment. And that's due at midnight too. Try not to get behind on those things. I know I mentioned this on the first day, but last quad, the biggest problem for people was that they said, I'll just finish it during the next block when I'm in my other class, and you won't. I mean, I don't know what you have in your other block. If you have power fit, maybe you will do it. I don't know. But certainly people said, oh yeah, don't worry, I just have chemistry. I'll just do it in the margins or whatever. And it's oh, you just actually never have time for that. And then it ends up carrying over, and then it stacks because then you start doing new things when you come back, and then it just turns into a crazy mess. So just don't do that to yourself. It moves so fast, and it's very unfamiliar for people because we just obviously never done this. So, or at least you've done it once, but depending on what classes you've had. So anyway, I have more time this quad. I actually have a prep period this quad, which is nice. So I'll try and get the marks for you as quickly as possible. You will technically get a mark after the first block that includes the assignments from the first block, but it's not really your mark. Like it's just, it is, but keep in mind that it's only just for the first block. So it's not really indicative probably of your progress at that point, but I will let you know. I'll let you know how you're doing and I'll post it. You guys already are aware, there's no exam. So how the course works is it's just all of the stuff you do in the course, and then at the end, that's your mark at the end. So there's no surprises or anything. You'll know exactly what's going to happen. Question?
Are there only 3 units?
No, there are 5.
Okay.
I only showed 3? That's my
I can't see the other 2.
That's my mistake. I forgot to turn them on. I'll do that right now. So there they are. If you refresh the page, you should be able to see them all now. It's diversity, genetics, animal structure and function, plants anatomy, growth and function, and evolution. Okay? Each one is pretty similar in that there's a major project that we do towards the end, often on the last day of the unit. There's a vocabulary list from the unit, and then there's a quiz at the end. Okay, I try and keep it as, simple as possible. I'm not trying to hit people over the head. We move super fast, so there's a lot of content, and I'm not trying to overwhelm people, but I try to keep it to that for each unit. And some people have been asking if the practice quizzes are worth anything. No, they're not. So they're for your own. It's like a knowledge check. You like do the practice quiz, and you're like, oh, I got 0 out of 10. It's like, whoop. Hold on, pause for a second. I don't think you picked up what you needed to pick up from that last little bit. Like you might want to go back and have a second look, okay? Or you aced it, you're like, oh yeah, 10 out of 10. I get this, I can organize these things into groups or whatever, I get it how it works. Then good. That's like a little check for you, just be like, all right, it's time for me to move on, I'm good, okay? So those are really useful. You don't have to do them because I'm not marking, but I really highly recommend them. And it's also a good, just an indicator to you to maybe consider emailing me or like I said, I will be in, I'll show you where you can find it. But so for example, if you look at the end of today's day 6. At the end of today, you'll see there's a little Google Meet right at the bottom, right there. I will be there for the last block for block 3, pretty much every day. I'll be there for the entire block. It's just drop in. So if you have questions, you're working on the project, you're like, I can't find a source for this kind of information. Whatever, really doesn't matter what it is, anything course related or even non-course related. You can drop by and say hi, but I'll just be there for you every day. So please take advantage of that. Nobody used it last quad. I probably saw 5 people the entire quad. And I definitely had students that needed to use it and just never did. You do not have to show your face. So I feel like one of the big barriers where people don't like to show their face, especially for the people at home, maybe you're like sitting in your room and there's like, it's messy or I don't know, whatever it looks like in your room. You don't have to show me video of your room, okay? Just you keep it private and you can just talk to me, okay? Voices are fine. And I won't make it weird, like I'll be voice only too, okay? If you want video, I'm just sitting right here, so whatever works for you. Other than that, everything's been going okay in general. All right, we'll try to make things a little bit more interactive now. We're gonna do most of the lessons live. I think it's about 50%. Some of them are pre-recorded, but a lot of it's live. And today's stuff is gonna be. So you guys left off. So day 5 is sort of like the switch over where it's like half one unit and half the other unit. So the first half was the project, right? Is that right? Yeah. The species spotlight assignment, and then the second half was a little review of organelles. This one I kind of squeezed into the second half of a day because I think this should be a review for everybody, right? Everybody's just like, oh yeah, mitochondria, powerhouse of the cell. Like everybody knows that stuff, right? I think. Maybe it was a little bit of a memory jog for some people. Not everybody knows what a Golgi does or whatever. But so this is sort of like the basis for most of the other stuff in genetics. It's like, okay, well, what in the cell does that? This thing. That's why we talked about it, so you know what that thing is, okay? Or like where it's happening in the cell. So today, we're going to talk about one of the key processes that happens inside the cell, and that is the making of proteins. They are like the functional units of life. And they're made by the things that are in the cell. We're going to talk about the Golgi, we're going to talk about the nuclear membrane, we're going to talk about ribosomes, because those are the things that do it. So it's good that you have a little bit of background on that. But nobody has any questions on the organelles or their function? Everybody feels sort of comfortable with that in general? Okay. Same thing for people at home, by the way, if you have these questions, don't be afraid to ask. I'm on just a tiny little bit of a delay, but I will catch your questions. Okay, get used to it a little bit. Okay. Well, let's get started then. So, yesterday you guys would have already watched. By yesterday, I mean Friday. I'm all mixed up here. Friday, you would have watched the overview video on how proteins are made, right? Like what is DNA and how does it work? Did you guys watch that? Hopefully. It was at the end of yesterday. I'll show you what I'm talking about here. It's this video right here. Yes. Great video. Great video. What we're going to do right now is talk about this process in a little bit more detail. And that this is sort of just like an addendum to that. The second one here talks about a little bit more detail about the structures that are involved, and we're going to talk about those as well. So these are really good overview videos. If you haven't watched them, you should watch them. They will really, really help with your understanding of this. Okay, so let's talk about it. We're going to do the little note together. You need part 2 of the course notes. It's right there. I will try in the future here to make sure that this stuff is all revealed in advance in case you want to print the notes. I don't think I did a great job of that here. Sorry about that. But in the future, I'll have everything laid out for you. So if you just want to go ahead and print them, if that's your thing, you obviously. It wasn't. Okay. Okay. Good. Yeah, and you can use Cami too. Is Cami working again now? I know it wasn't working on like the first day.
Yeah, it works for me. It
works. Okay. I did check on the first day and it said that they were having some kind of like server crash or something, but I think they fixed it at this point. So I paid for a subscription to it. So I'm hoping that it works. Otherwise I'm gonna be pretty mad if it doesn't work, but I think it is working. So anyway, so there's the course notes. So that's what you'll need. We're gonna watch this little video afterwards here. This video here is for the second part. So just to be clear, you guys have not done this yet. This is where we left off. I don't want to be like repeating myself just because I forgot to ask you guys if we covered this. We haven't done this. So we ended off talking about karyotypes, right? That's where we yesterday finished off?
Yeah.
Okay, cool. So this is pretty cool. This information is kind of important for the rest of genetics. So I'm just very quickly going to review this. You have 46 of these pieces of DNA. Yes, everybody's familiar with that? That's your 2N number, also known as your diploid number. Your diploid number is your full complement of all of your DNA. And for each of those chromosomes, you have a pair. They have the same genes on them. So for example, if we look at chromosome 1, you have one from your mom. Those are called maternal chromatids. And then you have one, or chromosomes, and you have one from dad. Those are your paternal chromosomes. And they are the same in that they have the same genes. So for example, there might be a locus on there for, a locus is just a spot, okay, a location, a locus for eye color, or there might be a locus on it for hair curliness, or there might be a locus on there for widow's peak, whatever. Did you guys, I can't remember what was in the previous lesson. Did you guys do that summary thing where you go through and talk about the Mendelian traits, the widow's peak and the, oh, good. That's coming still then. That's good, that's good. We're gonna talk about some Mendelian, some genetic traits. Anyway, they're the same genes, okay? But they're not identical to each other because the version of the gene, which is called an allele is a version of a gene, could be different from mom and dad. It could be the same. So maybe your mom gave you brown eye gene and your dad gave you brown eye gene. Or your mom gave you blue eye gene and your dad gave you brown eye gene. They could be different alleles, but the genes are the same. So that it's the same, it codes for the same type of thing, but it might not be the exact same thing. All right, so that's the key, is you have pairs. When you have pairs that have the same things on them, but not identical, those are called homologs, or you call this a homologous pair. That term is going to come up a million times in this course. We're going to talk about homologous pairs to death. So it's kind of an important concept here. But the homologous pairs have the same genes, but they're not identical to each other. So you got two sets of those, and if you break them apart into their two sets, your maternal set and your paternal set, those are your haploid sets. Okay? Haploid, or the 1N set, is half of your DNA. So if you look only at the maternal ones, that's haploid, 23. Or you look at the other 23, that's haploid. Haploid is half of the total complement of DNA that you need for an organism. So when we look at all this stuff that's coming here in a little bit, we're going to get into detail DNA replication and a whole bunch of stuff. Just keep that in mind that we're only ever going to show probably just like a couple of these. Like maybe we'll just show chromosome 1 and chromosome 2, and there'll be 4. But keep in mind that in you, there are 46 of these. So there's a little bit more going on here than we're ever going to show. Okay? In the next project that you're going to do, and obviously this is for genetics, we're going to talk about this a whole lot more before you get there. But it's really important to note here that there are 2 copies of each chromosome. Okay? They're not identical, but they have the same genes on them. All right. So I wasn't here for this, so I'm just double checking. So if we zoom in on any one of these guys, you talked about DNA structure already, right? Like the ATCG thing. We've covered that. Adenine, guanine, cytosine, thymine. Yes? Okay. So if you zoom in on these guys, it's a repeating pattern. Oh yeah, it's on the previous page here. Yeah, there it is. So it's a repeating. It's just a pattern of nitrogenous bases across the center. And you know what? I'm going to grab my giant DNA. Where's my giant DNA? Give me 1 second at home, guys. Grab my backpack. Giant DNA. Folks to be on the camera, just start in. What is it, this one?
No.
Yeah.
It's this one on the next page, yeah, personal.
What page is it? Maybe.
Find this page.
I'm looking for it.
Oh.
That's not our copy.
This is the one first page.
So there it is right there. Here, I'll turn the camera on so you guys can see this too. So you can see this thing. Is this gonna work? Maybe not.
Yeah, this is the first page. This is more of a first page.
I have one of those.
Yeah, so,
Yeah, that's yours.
yeah.
Okay, so this is the structure right here. This is when it's all twirled up into its double helix. And then there's actually another, there's another superstructure here, which is that once it's curled up into this, it's also wrapped up around little proteins called histones. So there's this, and then this cord is also wound up around histones. Not everything is wound around histones. So there's just a couple histones that are wound around each strand to kind of keep everything together. You guys last year talked about mitosis, right?
Yeah.
So at the very beginning of mitosis, you talked about how the DNA condenses and you can see chromosomes. Chromatids, rather. You can see it like turns into chromatids inside the cell. Does this sound familiar? So what's happening there is that it's getting wound around more of those histones. So it gets wound up. Basically, this pipe cleaner gets wound up around a whole bunch of other proteins and condenses all of the DNA into like little clumps, little logs. But in order for this process to work, this protein synthesis that we're going to talk about right now, this actually has to be kind of pulled out and unwound a little bit. Okay, so certain sections of the DNA, just sort of one section at a time, are getting opened up and unwound. And there are proteins that do that, that unzip it. If you take grade 12 bio, you get really deep into the biochemistry of that. What are the enzymes that stretch it out? What are the enzymes that unzip it, etc.? You don't need to know that for grade 11. So we're going to kind of do the basic version of this for grade 11. And then in grade 12, if you choose to take grade 12, where you get into a lot of biochemical detail. So the reason why we care about this so much is that this is sort of the key concept of biology, okay? This is often referred to as the central dogma of biology, the most important biological concept, okay? And the reason why it's so important is it's the connection between your code and you, what you look like what actually comes out at the end in an organism. That's a pretty important connection. We know a ton of stuff about DNA, and we previous to that, before we knew anything about DNA, all of our study was about outcomes, right? What do you look like? What are the shapes? So this concept is the connection between those two. It's how you go from a code to a shape. It's how you like make a thing out of the code. Okay? It's the most important thing. So if there's one thing you take away from this course, a basic understanding of how you go from a code to a thing is probably the most important thing that you learn this year, just as a heads up here. Okay, so this is a really important concept. But one of the other things to mention here that's really important for this is the idea that there are only specific things that can pair with specific other things. I already talked about this, I'm sure. But adenine always pairs with thymine because chemically, their bonding sites will only line up with each other. So if you had an adenine on one side here, the only thing, why don't I pick an actual adenine? There's an adenine. If you have an adenine on one side of the helix, there's got to be a thymine on the other side, okay? Chemically, they will not fit together. It's like trying to push two north sides of a magnet together. They will not link together if you have the wrong one in there, okay? And the enzymes that actually place them there when you're replicating your DNA, if they run into an error like that, they will detach. It won't allow it. Like you can't force them together, which is really, really helpful. It's like an error protection in your DNA. When you're making a copy, it prevents errors from accumulating in your DNA, which you don't want, obviously. You don't want random sequences to get put in there. So it does happen from time to time anyway. It's not totally foolproof, but it's quite a good system. So adenine always pairs with thymine, and cytosine always pairs with guanine because of their bonding sites. That's just chemistry. They only fit together a certain way. All right, so let's talk about how we actually make stuff out of that then. So we would have talked about that, I think, on Friday. The DNA has two sides, okay? So there is what's called the template side, and there's some extra information right here that you don't actually need to know. This 3 with a little 1 on it right here, that's the 3' end. It has to do with what side of the DNA is open, doesn't have something stuck to it. So if going back, way back here to this sugar down here, this sugar right here, it has nothing stuck to the 3rd carbon, that's the 3' end. And then the other one at the top has nothing stuck to the 5th carbon. Or sorry, I think I have that exact opposite here. This one has something stuck to the 3rd carbon. This is a phosphate. And this one doesn't. This one has nothing stuck to. No, that's not right either. Hold on. What do I have wrong about this? There's an error in this diagram. That's my problem. Here, this one makes more sense. Here, there's nothing stuck to the 3', and here there's something stuck to the 5'. Okay? Anyway, the chemistry of this isn't important. You don't need to know that. But there's a directionality to the DNA. Okay? In other words, when you read the DNA, when the enzymes make copies of pieces of the DNA, they always read it in the same direction. It's sort of like having a book with a front cover and a back cover, so you read the pages in the right direction. If there's no directionality to a book, one person would read it front to back and the other person would read it back to front. One of those ways doesn't make sense, right? So it's the same thing for DNA. It only gets read in one direction. that actual information, knowing which side is which, is not something that you need to know in this course. I don't even know what I have in my note. Someone might have asked about it last time. But as long as you know that you need to read it in a certain direction, the actual direction doesn't matter. You don't need to know that in grade 11. Grade 12, someone will ask you to know that, but it doesn't matter for now. So there's two sides here, okay? There's the template side, and then there's the coding side. So the coding side is the actual code, okay? That's what you want to make a copy of when you make your protein. In other words, you're going to build an mRNA, which you saw in the video on Friday. That's your message to the ribosome, to explain to the ribosome how to build the protein. And you want that message to be the same as the code, the coding strand, okay? That's your actual code. So the other side, the template, is sort of just like the backup, okay? It's not the code that you want at the end, and you use the template to make a copy of the code. Just like if in art or whatever, if you made a template for something, that would be what you would use to make your final product. You use the template to make the final product, right? The same thing is true for DNA. So the template strand is what you're going to use to make the mRNA, but at the end, you want it to be the same as the code. The idea is you're making a copy of the code using the template. And it's a really cool mechanism here because it just relies on chemistry in order to make this message that you're going to send. Which is, I mean, everything in the cell relies on chemistry. It's cool that the chemistry is so foolproof. Like it's like an error checking method of chemistry to make something. The organization of life is actually just insane once you really get into it. But this is anyway. There are two basic processes here. Okay, the first process is the making of the message. Okay, the message, which you're going to deliver to your ribosomes, those are the builders, is called mRNA. And the process of making that message is transcription. Okay? Transcription is making the message. That's the first part. So you got to make the message. You could also think of it as copying part of the DNA, just like you would make a copy of a page from a book. Okay? And an identical copy, you're just going to copy it. Making an identical copy of a page is called transcribing a page. That's what it's called in English. And that's exactly what this is. It's just making a copy of something. Okay, so transcription is the first part, transcribing the DNA. And then you got to take that page and then follow the instructions in the page. Okay? So you're taking a message and making it into a thing. That second part is called translation. So the two parts are transcription and translation. Transcription means to make a copy, and translation means to make it into something else. Now you're going to make a message into a thing. That's the second part. Okay, so let's go through how this actually works. You've got your DNA strand. An enzyme is going to come along and it's going to unzip it. Okay, it just runs down the center and it separates. That's what my orange line is here, by the way. It comes through and it just whoop, cuts the DNA in half, usually only in a certain area of the DNA. Again, you don't want these unbonded ends of nitrogenous bases just sticking out all over the place because it makes them vulnerable. They can chemically bind accidentally with all kinds of things, right? That's also, by the way, the reason that this stuff is all in the nucleus, because the nucleus is like a safe zone for DNA. There's not just stuff floating around in the nucleus. It's only stuff for copying DNA and replicating DNA, and that's it. And ribosomes, which are produced partially in the nucleolus. But that is the only thing that's in the nucleus. So you don't have like reactive oxygen species. You don't have sugars. All kinds of things that are prone to being chemically reacted. Okay, so you kind of keep all of those different reactants for chemical reactions out of your nucleus to keep the DNA safe. Okay, it's like a little bubble of protection around it. So you unzip it, and now we want to make a copy. The coding strand at this point doesn't do anything. Okay, so you can think of the coding strand as kind of being isolated. It's basically uninvolved now in the process. It is the code, it's what you want to make a copy of, but you don't use the coding strand, you use the template. So what happens then is another enzyme is going to come across, and it's going to build a complementary half to the template. It's going to zip along and go, okay, what is supposed to stick to T? What sticks to T? A. Adenine sticks to thymine. And then it comes to an A, and it goes, oh, what sticks to this A?
Thymine.
Normally it would be thymine, okay? One small exception here is that there's no thymine in mRNA. It doesn't exist. So mRNA has one different base pair, so every thymine is instead a uracil, okay? It's chemically almost identical to thymine, but it has one different chemical bond in it, making it a different nitrogenous base, okay? Just adding in a little bit of extra complexity here, but all the T's become U's. Okay, so you're not going to see any T's ever in mRNA. If you're looking through and you see a code and it has a U in it, it's mRNA. It's not DNA. Okay, so we got a U, C binds with, just throw it out there, G, G binds with, C binds with, G, G. This would be a T, except there are no T's, so it's a U. T, A, G, C, A, U, T, A. Okay? And again, this is just chemistry. Only one of these things will fit. So as it's going along, it's going to grab the nitrogenous base that sticks to it. And if it doesn't stick, then it's not going to stick. So you're not going to end up with it there. That is the building of your mRNA, the messenger RNA. It's only single-stranded. So because it's single-stranded, this piece can fit through the tiny holes in the nucleus, the nuclear pores. Okay? So this little piece is going to get stuck, and then it'll detach, and it'll get carried out through the nuclear pores, and then it will end up in the cytoplasm. Okay? That's the making of the message. And you'll notice that it was completely built on the template. So the template builds the message. Everybody with me so far? Okay. So we took that template strand, we went along, we added in all the complementary base pairs, and then it detached, and now we had this stick, this message of how to build a particular protein. Okay? One of these sections is called a gene. Okay? A gene codes for one specific protein. So we usually only encode one protein at a time, so that's one gene is being read at one time. You got about 50,000 of those sections, okay, interspersed all over your DNA. That doesn't account, by the way, for most of your DNA. Most of your DNA is actually just there to control when to make copies of various things. So it gets wound up in various patterns, just so your eyes don't start producing hair and things like that. You know, you gotta make sure that the right cells are making the right things. So all the rest of your DNA is directed towards controlling which things are turned on and which things are turned off. Okay, so now we've got this piece of mRNA. We've done the transcription part. We got to make it into something. So the first thing, let's start here by what actually does the translating? What makes it into a protein? What's the organelle involved? Anybody remember? You guys went through all the organelles on Friday. What one is the one that reads the DNA and actually makes stuff? It's the builder of proteins.
Amino acids?
So amino acids are what it's being made out of. You're right, they're in there. Those are the building blocks. You're right. Those are the Legos that make up proteins.
Yeah.
But something's actually building something out of the Legos, which is what? It is the ribosomes.
Oh, yeah.
So the ribosomes are also made out of RNA, blah, confusing, but they're made out of a different type of RNA called rRNA, ribosomal RNA. That's why they're so small. So when you guys were doing your diagrams of cells, you may have noticed the ribosomes are basically just dots. And that's because they're made out of RNA. They're just made out of tiny little pieces of RNA, so they're not actually that big. They're quite small inside the cell. So, and a ribosome is going to come along. It's going to hook up to this RNA, this other type of RNA, mRNA, and it's going to start reading it. But you have to know where to start. So just like when you read a book, there's like a word that's like your first word in the book. It's like, and you know that because it's like chapter 1, word. Okay, right? There's like a first word there. Anything that comes before the chapter 1, you don't have to read that. That's like introduction. It's like telling you where it's published and stuff like that. The ribosomes don't care about any of that stuff. They don't care about that part of the story. They only care about what comes after you say chapter 1. So the chapter 1 for cells is this. It's always the same, at least for humans it is, I should say. Other organisms have different start codons. But it's a group of 3 letters called a start codon, and that is AUG. So when the ribosome sees that, AUG, that's like the same thing as chapter 1, okay? It's like, whoop, this is the beginning of this code. There's a section before that usually that's just nonsense. It's just, like I said, it's just like a bunch of letters, and it doesn't read that part. But then once it gets to AUG, it's like, all right, it's go time. This is where we start translating right at this chapter 1 indicator. Okay, so that's the start codon. The start codon also codes for an amino acid, as you mentioned, which are the Legos, the building blocks that you make this stuff out of, okay, the smallest subunits of proteins. And the smallest subunit that it always starts with is methionine. So methionine is the very first amino acid in every single protein that you make, which is weird. It always starts with the same piece of Lego at the beginning. Okay, I use Lego as an example here a lot because my son is obsessed with Lego. But we insert whatever your building piece of choice is here. But they always start with methionine. So that's the start codon. Then it goes across and it reads 3 letters at a time. It's key, it's got to be 3 letters. And every 3 letters will code for a different amino acid that the ribosome will grab out of the cytoplasm and hook up to the end of the line, okay? And as it grabs 3 letters, 3 letters, 3 letters, 3 letters, eventually it's going to start building this polypeptide, this long sequence of amino acids, okay? So I should mention here that at this point it's not a protein yet. So that's why I call it polypeptide. A polypeptide is just a whole bunch of amino acids strung together. Once you take that big long string and you fold it into its final shape and it's functional, then it's a protein. But before you fold it up and make it work, it's basically just a whole bunch of things strung together. That's a polypeptide. So it's the difference between, I'm sure you guys have made an egg before, okay? When you crack open the egg, those are proteins, okay? But when you cook them, you actually unravel all of the folds in those proteins and they turn white. That's a polypeptide, okay? You're actually breaking down the proteins a little bit when you cook it. And once they unfold, they're no longer transparent any longer. They turn white. And now you're looking at a whole bunch of polypeptides instead of proteins. You've broken them down a little bit. They're unfolded by heat. You guys have a method for doing that as well, the acid in your stomach, and we'll get to that later. We'll talk about how that works. But that also unfolds them. It denatures them. The word is denatures for protein. Okay, so we've got 3-letter sequence here. Start, and then you go along and you just read these codons as you go. So the next one in this particular one is CGG. We've done a great job here, and we've figured out the whole code. Very useful. We know what amino acids go with each part of the code. So you can take any part of DNA and you can figure out what does this make, which is pretty cool. It's led to us being able to predict a lot of the time what specific genes do. Because we can just use a computer, we'll go through and it'll just say it'll make this sequence. And then you can use another computer, a big supercomputer, that will predict how that polypeptide will get folded into a finished shape. And then sometimes you can guess what the chemistry will be, what that shape will do at the end, if you use a good supercomputer, okay? If anybody's ever, probably nobody's ever done this before, but you can get a little program to run on your PC. That might be for Mac too, I don't know. Anybody heard of folding at home before? Nobody's heard of this. Anyway, what it is a crowdsourcing program for figuring out what shapes, what proteins are going to be made from a given DNA sequence. And what they do is they send little bundles of practice to different computers, and they crowdsource it, and everybody does a little bit of the math and then sends the math back to some central computer that puts it all together. And then they can use that to predict what shapes will be made from these proteins called folding at home. Anyway, I'm so nerdy. When I was in high school, I had this on my computer. I thought it was the coolest thing. That was way back. It works a little bit different now, but it's very similar. Anyway, so how you read this thing is you start in the middle, okay? So your first letter is the letter in the middle. It's going to be 1 of 4. In this case, well, we already did the start codon, AUG, but let's do the second one here, CGG. Okay, so CGG would be, let me erase this. Get out of here. C, and then it travels out from the center. So CGG. So CGG gives you arginine. Arginine is an amino acid. Okay, that would be the next one that gets linked on. And then this one gives you the short forms too, which is nice. So ARG is the short form for arginine. You don't need to write out the whole thing. And so then that would be the next amino acid in the chain, arginine. Okay, and then it goes on and reads the next codon, whoop, UAC, and you can look that up. UAC. Okay, that's tyrosine. Tyrosine is TYR. Feel free to use the short forms, but there's the next one. Okay, so then it attaches the tyrosine on there. And then you read the next 3, UUA. Change my color here. UUA, and UUA is leucine. So then it's going to hook up a leucine. Okay? And this goes on and on and on and on. It'll keep reading the mRNA until it comes to a specific part of the code that says, you're done. Okay, that's called a stop codon. So the stop codon is the opposite of a start codon. It tells the ribosome, this is the end. This is where the protein stops. Okay, so you can stop putting extra things on. Okay, so the stop codons are all marked on here. There's one right here. That's a stop. That's a stop. There's more than one stop codon. And these are specific for humans. So I should mention that, or mammals actually. It'd be the same for all mammals. Once you go outside of our phylum, sometimes these are a little bit different, but they're often similar. So at least for all mammals, these are the same. So, so for example, this stop codon would be UAG or UAA, and then this one right here is UGA. So those would be the 3 for mammals that say, okay, stop coding this. This is done. And once that's done, that pops off, and then you got to send that polypeptide that you've just assembled, so this big long thing, you got to send it over to something to do some folding. So some of the ribosomes are attached in the cell to a structure that already does that. That's sort of like the finishing folding plant that does the end. Anybody know what that is? There's one of the organelles has ribosomes stuck to it. Oh, do you remember what this say? It's okay if you don't. Do you remember what it is?
The rough endoplasmic.
The rough ER. Okay? So if it's attached to the rough ER, those ones are going to get fed through the rough ER. Okay? In fact, it actually builds the protein into the rough ER. And it does the folding. So the rough ER is going to do some finishing touches. Sometimes it snips off little bits of the polypeptide to make everything into like a neat little package that folds properly. Okay? That is how you package up proteins that you're going to send out of the cell. Okay, so the rough ER is used for processing proteins that are going out. In other words, let's say that you are a cell in the pancreas and you make insulin. Okay? Those are called islet cells, by the way. That's their job. Pretty much their only job is just to make insulin. If people are not familiar, insulin is a hormone, and every time you eat something that contains sugar, you release a little insulin, and it tells all of your body cells, take up that sugar, eat it. So that sugar will go into your liver and your muscles and stuff like that, so it can use the sugar. If you don't do that, if you don't produce insulin, the sugar just stays in your blood and can cause a lot of problems. It's called diabetes. So there are cells that just do that. When they produce insulin, they do it through this process. So they make it into the rough ER, and the rough ER will send it to a Golgi, which, do you guys remember the function of the Golgi? Do you remember?
Package proteins that do themselves.
Exactly. So it's a packager. So it takes those proteins that have been made into the rough ER, puts them into a package that is ready to be released out of the cell whenever that particular protein is needed. In other words, in this case, in the case of insulin, it would wait until there's sugar in the blood, and then it would just push that little package out and be like, here's your insulin. It's ready to go. It's sitting in this package. That's the job of the Golgi. If you are building a protein for inside the cell, so let's say you're building a protein that's going to be a muscle fiber, or you're building a protein that's going to build other cell organelles, or you're building a protein for communication with the outside of the cell, a transmembrane protein, or you're building a protein that pumps stuff into the cell or pumps stuff out of the cell, or whatever, okay? Those are called intracellular proteins. If you're building one of those, that's what the ribosomes in the cell that are just floating around, they do that, okay? So that's the easier process. If you build it, if the ribosome that's doing this is just floating around, all of those proteins stay in the cell. They're not being packaged for export, basically. So the free-floating ribosomes build the intracellular proteins, and the ribosomes that are attached to the rough ER build the intercellular proteins. Inter means between. Now, I can't remember if I mentioned this anywhere on any other note, but did I talk about the difference between inter and intra? Did that come up? Okay, that's going to come up in biology about a bajillion times. So I'm going to just note it here at the top of the page.
Is it between within or something?
That's right. Exactly. So inter means between. So if you're talking about intercellular proteins, you're talking about proteins that are going between the cells. Okay, they're outside of the cells. In biology, if you ever see intra means within. So an intracellular protein means a protein that you're building for in the cell, within it. Okay? This is used in biology all the time. This is constantly being used, and unfortunately, they sound very similar. So sometimes you just hear intracellular. You have to be like, oh, is that intra or inter? It's a big difference, okay? So intra and inter, one is within and one is in between. Okay, sometimes you'll hear about intercellular signaling. That's signaling between cells, and intracellular signaling is sending a signal inside of the cell. Those are two completely different concepts. All right, so we talked about this code, how you can decode it with your sweet decoder ring. I just want to quickly mention how the ribosome is actually doing this, because it is kind of cool. So here you got your mRNA strand, and it's sandwiched in this ribosome, which is made out of rRNA. And as it runs through, what it's using are these little guys. They kind of look like little, I don't know what to call that. What would you? Like little T's, I guess? Or little clovers. I think sometimes they're called in the text, but they're called clover shaped. They're not really clover shaped, but what would you call that? Some kind of like weird stick figure? Anyway, that little stick figure is called tRNA. It's also made of RNA. And this is what actually brings the amino acids to the ribosome. So these are floating around inside of your cytoplasm, and there is a different version of this for each amino acid, and they have a code on them. So the code is complementary to whatever the code is on the RNA. So this would be the same as the template strand, this code, right? Complementary to the code. And they have that complementary piece on one side, and on the other side, they're actually holding on to the amino acid. So this is the thing that is actually getting physically brought in and attached. Once it gets attached, that little amino acid actually gets kicked off, and then this tRNA just kind of whizzes off into the cell empty, and it will find another amino acid and bind to it so that it can be used over again. It gets recycled over and over and over and over again. Okay, so that's how the process is actually happening. It's all chemistry, which is kind of cool. And then as it's doing that, it's dropping them off. There's a bond, and then your protein kind of grows out of that. Okay, I realize that that's a lot of information. You guys saw this in the video yesterday. So I'm going to go through the process one more time. Okay? So, you start off by making a copy of a piece of the code. Okay? So you unzip the DNA, and an enzyme is going to come along, and on that template strand, it's going to build a complementary strand. All the complementary base pairs, T, A, G, G, C, T, T, A, D, G, G. Maybe I'll show you a video of this later. It happens so fast in real life. So these enzymes can zip across a piece that's maybe 200-300 base pairs long in less than a second. It just goes whoop, and then a little piece of DNA flies out of it. It's really cool. So they make that little strand, and then that's the transcription part, making a copy, and it sends that out of the nucleus, okay, out of the safe zone and into the crazy chemical soup of the cytoplasm. Once it's out there, it's going to get attached to one of these guys, a ribosome. And that ribosome is going to look for a start codon, also codes for methionine, okay, AUG. Once it hits AUG, then it's going to start grabbing amino acids out of the cytoplasm and adding them 3 base pairs at a time. This is the translation part. So we've got this base pair, then this base pair, or not this base pair, sorry. This codon, then this codon would be this amino acid, this amino acid, this amino acid. Next, 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 until it hits one of the 3 stop codons, which are UAA, UAG, and UGA. If you guys don't know what I'm talking about here, I'll highlight them all the way down here. So we've got UAA, UAG, and UGA. Once it hits a stop, that's the signal to let go. If it's attached to the ER, then the ER is going to do some folding. It's going to help sort of configure that polypeptide that you've just built. Lots of new vocabulary here probably for you. This long chain of amino acids. And at that point, sometimes it's a protein at the end there. Sometimes the Golgi has to do a little bit more modification before it becomes a final protein. But anyway, the ER is going to do a little modification. Snip, snip, does some cutting, does some folding, and then it gets sent along to the Golgi, which is going to put it into a vacuole. It's going to put it into a little package, or a vesicle, I guess. It's not big enough to be a vacuole, but it's a vesicle, which is just this little bubble basically made out of fats. And then it's ready. Anytime you need it, you can just kind of push that to the edge of your cell, and it'll bind to the cell membrane and just express the proteins out into the intracellular fluid, into the blood, whenever it's ready. So the rough ER, they receive a coded message from the nucleus. Okay, that's mRNA. The mRNA gets fed through a ribosome, and I had to make a correction here. It's not a protein at that point. It's a polypeptide. So polypeptide is a whole bunch of amino acids stuck together. Then the newly created polypeptide, not a protein yet, is going to move through the rough ER and get pinched into a vesicle. So a vesicle, again, is just like a little bubble that has a membrane around it. It's a membrane bubble. And that's going to go to the Golgi. Somebody asked me last year what the difference between a Golgi body and a Golgi apparatus is. They are actually different things. A Golgi body is only one of the pancakes in the Golgi apparatus. So when you see the Golgi, you know how it looks like a bunch of blobs that are kind of next to each other? Each blob is a Golgi body, and then the whole thing together, all of the blobs together, is the Golgi apparatus. So that's like a little...
So would it just be like one Golgi body would take in like the protein and then package it? Or would it be like it goes through all of them?
I think it's just one. It is just one, yeah. I think they're identical. They're all identical and just linked together, yeah. Okay, so once it gets to the Golgi, that's when it really gets folded into a complex shape. So the Golgi is doing like sort of the final packaging of this thing and making it functional. One of the reasons why you don't want to do that until the very end is because if you make the protein functional, it's going to start working. So if you're transporting it around the cell, maybe you don't want that protein to start being functional inside of your cell yet. It might start doing chemical reactions inside your cell that you don't want to happen. So you kind of wait until right at the end of the line to just do some final little activation, finagling on this protein, and then that makes it active, so it's ready to go. And then it's once at that point, it's bound inside of a membrane. It's not going to interfere with anything inside of the cell. So now accidents happen, but in general it won't. When we talk about this stuff, we're always talking about like best case scenario. Yeah, sometimes the cell makes mistakes. Sometimes the proteins get made incorrectly. Sometimes they accidentally get released in the cell and cause apoptosis and the cell will lyse or something like that. So the cell might accidentally kill itself or something. Like that stuff happens all the time. But this is best case scenario. This is what you're shooting for. It's not perfect, but it's pretty darn good. So at that point, once you've actually folded it up into its correct shape, now you call it a protein. So in all the stages before this, you wouldn't really call something insulin. It's like pre-insulin or proto-insulin. But at the end, once you fold it into its correct shape, then it's insulin at the end, because then it actually does the thing, whatever its function is. Okay, and then it hangs out in a little vesicle. Again, it's put into a little membrane-bound package. And eventually, when you need it, you can just dump it into the extracellular fluid, which is continuous with the blood. It's not the same thing as blood, but it's the liquid water part is continuous with the blood. It goes in and out of the blood. So there's no red blood cells in there, but it's water with some salts and things dissolved in it. So to be clear here, we're talking about protein production for an intercellular protein. If you're looking for an intracellular protein, you just have to cut this off earlier, okay? Right when the ribosome makes it and it goes out into the cytoplasm, it's done. That's basically it. Sometimes there's a little bit of post-folding that goes on inside the cytoplasm, but it's pretty much done at that point. So this is intercellular protein production. So I realize a lot's going on here. Oh, I have more down here. Oh yeah, this is just where I mentioned this idea that the rough ER is contributing to extracellular proteins. Yeah. Or intracellular. Oh boy, now I'm mixing it up. Intercellular protein. This is, like I said, this is the connection between genetics and all the rest of biology. So later on, we're going to talk about, enzyme production and like what they do and systems, and all of that stuff is regulated by your genetics, and the connection between the two of them is the protein synthesis. So in the latter half of this unit, we're going to be talking about how a certain code will give you, hairy knuckles or whatever genetic trait. And this is the connection between the two of them. So it's a different code, means different proteins, means different physiology at the end. Or phenotype, the word is phenotype. We okay with this? Yeah? Okay. A little practice. Okay, I got my finished copy here for you. We'll go to that in a second. 2 things I'm going to get you to work on right now. Let me just double check here what I've got in the second half. More this than I intended. That's okay. Self-review questions on the last page. Ooh, busy day. Okay, I'm gonna give you a while to work on this. So there's 2 things I'm gonna get you to work on here. So one of them is I'm gonna get you to go through here and create a polypeptide for each of these codes, okay? So they work, each one of these is a little bit different. The first one starts by giving you a DNA strand. Keep in mind that the strand that it's giving you here is the template, okay? So it's giving you the template. That means that when you build your mRNA, you're going to build it on the template. So what I'd like you to do is build the mRNA on it. So it gets built right on the template strand, which is the top strand there, which would be what? What goes along with T?
A.
A. And then A goes with
Well, U.
U. Nice. C. Okay, and then C is G, C, G, etc. You guys get the idea, right? Okay, so on the template, you're going to build your mRNA strand. I'm also going to get you to build the coding strand. The coding strand, it's not actually marked on yours, but you can just add it in there. Right underneath and attached to your DNA is the coding strand. That has the actual code on it. That is going to be complementary, but keep in mind that there's no uracil, right? So this is just another piece of DNA that's going to be attached on the other side. So in other words, T is going to bind with A is going to bind with T in this case, okay? C is going to bind with G. So you're going to show me what the coding strand would look like, okay? And then you're going to show me what the mRNA sequence would look like. And then you're going to use this little tool right here to figure out what the polypeptide is going to look like. So that's the AA stuff at the bottom, amino acid sequence, okay? AUG, that's the start codon. It's pretty much always going to be the same for you, and it's always methionine. Okay, so I'm kind of cheating here, but methionine is always the first thing. This is a shorthand for it. So methionine is first. So that's for the first one here. For the ones below here, sometimes I'm going to give you the mRNA, and I want you to build the DNA. On the top, we're always going to do template on the top and coding on the bottom. There's no actual convention for this in terms of like, do you do the coding strand on top or the template below? But just to make things simpler for this class, I'm always going to do the template on top and the coding one below. Exactly the same as we did way back in this example right here. Okay, where the template's on top and the coding one is below. I'm going to keep it always consistent, otherwise it starts getting confusing. Which one is which? So I'll always put the template on top. So you guys are going to do the same thing. So you're going to show me, for this mRNA sequence, what is the coding strand, what is the template strand, template coding, template on top, coding below, and then what is the amino acid sequence that you get from that. Okay, so you're gonna get you to work backwards, and then I'm gonna get you to work backwards all the way from the amino acid sequence. An important thing to note for this one is that for a lot of these, there's actually more than one mRNA sequence that makes that amino acid, okay? Whichever one you use is fine. As long as it actually makes that amino acid, it doesn't have to be exactly the same as mine. It's totally fine as long as it's the same. The only one that has to be the same is methionine, because you have to use the start codon at the beginning. That's the beginning of every gene. You have to use the start codon. But other than that, because there's more than one that codes methionine, but for all the other ones, it really doesn't matter. Okay, if there's like 3 different ones for arginine, just pick one. Any one is fine. Okay, and then the same thing going on here, except this one, I give you different ones. So I've given you a piece of DNA and a piece of mRNA, and I've given you some amino acids. Okay, it's a mix. So sometimes you're gonna be working from the bottom, and sometimes you're gonna be working from the top. And by the time you're done this, I think you'll feel very confident, I hope, in how to read this, how you actually read the code and convert it. Okay, these don't line up perfectly. Do your best. They're obviously in codon, so they're in groups of 3. So for some reason, mine came out really well as a PDF, and then I thought I gave you the same PDF, but some of them came out funny. So I'm not sure exactly what happened there. Second part, once you've done that, is I'm going to get you to go through and write out the process in your own words. Tell me what's happening. You can use some of the terminology, absolutely, that we just used, but don't look it up. I want you to write out step by step, how do you go from a code, from a piece of DNA, to a protein, and let's talk about an intracellular protein, one that's going to leave the cell. Okay, we may as well learn the whole process from beginning to end. All right, in your own words. I would number the steps. That makes it easier. And so what I did is I just numbered it on my diagram up here. And so it's like, well, what's everything that happens at this location? What's everything that happens at this location? These are the ribosomes that are attached to the ER. What's everything that happens in the Golgi? What's going on here? Okay, so I would recommend kind of sort it out like this, kind of step it, but I'll leave it up to you how you want to organize it. All right, I'm going to give you a while. Let's work on this. If you're doing this, if you're going through here and you're like, I don't understand what this is. I don't know what's going on right here. Call me over, okay? Let's chat about that. So make sure everybody's up to speed on this. Same thing for people at home, okay? Okay, I'll give you guys a while to work on this. Give it a shot. Hey guys at home, if you wouldn't mind in the chat, just letting me know that you're here again. I got to take attendance again. For you guys in the room. Is this gonna work? Yeah. Oh, it's a little bit on a delay. Yeah. Oopsie daisy. Sorry guys. Why is this? Oh, I see. Gotta do this. There we go.
Why? I apologize. You should apologize. This is my top 2, right? Yes.
Nolan, you're here now. Russ. Okay. Quickly restart this. Okay. First of all, before we keep going here, does anybody have any questions from the protein synthesis stuff from before the break? Everybody's okay with that? Okay. So like I said, that's sort of like the basis of what we're gonna It kind of ties into everything that we do for the rest of the course, evolution, genetics, all that stuff. This is going to be a review for you, I hope. You guys did mitosis last year? Yeah. So it is possible that you did it during the, I don't know what you call that at the end of the year last year, the dark phase, where I'm not sure how carefully you covered mitosis, so maybe not great. So we are going to go through everything again, but I do go through it faster than I typically do. If you're one of those people that has never seen mitosis before, because maybe it was something that you didn't have time to get to during that time, let me know, okay? This may require a little bit more review than is typical. So in mitosis, the basic purpose of this thing is to make a copy, okay, of everything. So everything gets copied to make identical daughter cells. And this is for adult, otherwise known as somatic cells in eukaryotes, okay? So more specifically here, this is actually the eukaryotic cell cycle. Oops, that's not the letter I'm looking for. So if you'll remember from the last unit, a eukaryote is something with a nucleus and organelles and et cetera, okay? So it's the kingdoms that are not bacteria and archaea. Bacteria and archaea do something similar to this. Binary fission, which we did talk about. That was in the last unit, right? I don't have a laser memory for it, but yeah, okay. So binary fission is like this, but it's missing a lot of the structures and sort of the exact things that you see in mitosis. For example, there's no centrioles. There's no dissolving of the nucleus. There's no nucleus. There's no chromosomes because prokaryotes don't have chromosomes. They just have like a long single strand or a loop of DNA inside. So a lot of this stuff is missing. It's a similar process though where you make a copy of everything, separate it, and then split the cell in half. But again, it's not exactly the same. So what we tend to focus on here is just the DNA component. So based on what we were just talking about before the break, that's really the most important part. Yeah, you got to make a copy of your Golgi and you got to make a copy of your mitochondria and whatever. But the DNA, the instruction set, is the most important part. If you're missing a Golgi and you have the instructions, you can just build another one. But if you're missing the instructions, your cell is toast. It has no chance of surviving once you've done mitosis. So we're really just going to focus in on the DNA component of this, which I think is what you did probably in grade 10 as well, right? You just talked about separating out the chromatids. Yeah. Okay. So it's important to note here, and I'm sure you discussed this last year, that most of the cell's life cycle is interphase. Interphase is the between mitosis phase. And that's the part where the cell is doing its job. So for example, going back to what we were talking before the break, if you are a pancreatic cell, that's the part where you make insulin, okay? Where you're just doing your job, being a cell, making insulin. Or if you're a rod cell in your eye, on your retina, you are receiving light signals and transmitting them as electrical signals down a nerve, or whatever you do, okay? Or if you're maybe a hair follicle, you produce hair. Whatever you do as a cell. So interphase is the most important part, and in general, almost all of your cells are in interphase, except for a small percentage. So things are typically in interphase, and most of your cells are in interphase. If they're not in interphase, and you also talked about this last year, you guys talked about cancer in grade 10?
Yeah.
Okay, a little bit. So anyway, if you have a lot of large grouping of cells that isn't in interphase, it's probably indicative of the fact that there's something carcinogenic happening there. Anyway, interphase is broken into three parts. So the first part, the G1 phase, is just growth and organelle building. The second part, and for our purposes here, when we're discussing replication, that's the most important part, is the part where you make a copy of the DNA. By the way, you may be wondering why we're reviewing this. So the reason we're reviewing this is because right after this, we're going to talk about meiosis, which looks like mitosis, but the purpose of it, instead of making adult cells, is to make gametes. Gametes are going to be an egg or a sperm, so for making offspring. And that's where we're headed to next. That's the one of the main sort of thrusts of this course is sexual reproduction, how you take all of your genetics and only take half of it. Just grab half to make a baby because you can only contribute half, right? And then a partner would contribute half. So it's very similar to this, so you got to make sure you kind of have the mitosis under your belt first. So anyway, G1, the cell's going to grow, it makes proteins, builds organelles, then you double the DNA in the synthesis phase. In chemistry, you guys talked about synthesis last year, grade 10. That's a building chemical reaction, it's exactly the same thing. And then the last part here, the G2 phase, is where you start making some structures that are used for dividing, usually centrioles. So centrioles are produced there, and there's a little bit of growth that happens in the cell as well, but it's mostly centriole production. Then you got your mitotic phase. That's the part where you separate the DNA out, your 2 copies to either end of the cell. And then the last part here is cytokinesis. Cytokinesis and mitosis are actually separate, and cytokinesis starts before mitosis ends. So there's actually like an overlap between mitosis and cytokinesis. Sometimes if you like watch a video about mitosis, they talk about cytokinesis like it's a step in mitosis, but it happens kind of simultaneously with mitosis, and it's biochemically separate. So this is not exactly the same thing, and it happens after mitosis is over, or starts anyway before mitosis is finished. Okay, so that's the basics. So some of this stuff we already talked about last year. I don't know how much of this will be review. I should have gone back over that note. You guys already talked about the maternal and the paternal stuff, right? We even reviewed that at the beginning of today. So you've got a set that looks like this, 46, and you got to make an identical set of 46. Once you've done that and made a identical copy, it's going to look like this. By the way, I don't know if I actually managed to delete this part out of your X chromosome at the bottom. If there's 3 there, you got to put something over the last one. There should be only 2. By the way, male or female?
Female.
Female. And you know that because?
There's 2 X.
2 X's. This person actually had 3 X's, which is an anomaly, which we'll talk about later, but 2 X's is typical. So you notice the only difference between this and this is that now each chromatid, chromosome, has a copy attached to it. I'll try not to mix terminology up too much here. They're called chromosomes in all of these contexts, okay? So these are chromosomes, these are chromosomes. When they're unraveled, just sitting out as chromatin in the cell, even before they become these X shapes, they're still called chromosomes. So they're chromosomes always, but when we talk about chromatin and chromatid, those are 2 different things. So chromatin is when the DNA is just laying out in the cell and you can't actually see any structure. So that's when it's unwrapped, basically. And the reason it's unwrapped is because it has to be easy for you to do this. Make copies of sections and stuff like that. If it's not unwrapped, it's super hard to get in there and make copies of little parts of it. So normally it's chromatin and it's just unwound and laying out. But later, at the beginning of prophase, you're gonna wrap them up into these sausages called chromatids. So the main terminology mix-up that happens here is the difference between these, like for example, if we look at chromosome 1 here, these 2 things, and these, okay? They look similar, but we're talking about totally different things. So 1 and 1 here, this is chromosome 1 from dad and chromosome 1 from mom, maternal and paternal. Those are homologs, right? They have the same information on them, but not identical. Once you make a copy of each and you end up having this, so you have 2 from dad that are identical and 2 from mom that are identical, these are called sister chromatids, okay? That is, the copies are called sisters. They're sisters to each other. The homologs are still there, so you have chromosome 1 from mom and chromosome 1 from dad. Those are homologs, but within the same one, those 2 things are called sisters. Okay, so for example, if we just looked at the blue ones here, those would be sisters, and or you just looked at the pink ones, those would be sisters. They're sisters to each other.
Are they always identical?
I mean, as long as you, during the synthesis phase, as long as you made an identical copy, they're
So
identical.
the majority of the time they're identical?
Almost always, yeah. Their errors can be made, so there's not always identical, but the idea is to make them identical. Yeah.
Okay.
You've made a mistake if they're not identical. Now the pink and the blue one here are not identical though, right? Those are homologs.
Yeah.
Yeah, okay. But within the single one, they're identical. So that difference between homologs and sisters, I think, is the number one thing that confuses people, is people confuse the idea of sisters and the idea of homologs. So the homologs are like the two, one from your mom and one from your dad, that are called chromosome 1 or called chromosome 2 or called chromosome 3. And then when you make copies, the copies are sisters. So you got to really separate those two ideas out in your head. So you've made copies of each, okay? You do that at the beginning or during the synthesis phase. And then the rest of the process is all just separating them out. You got to put one sister on one side and one sister on the other. That's what you want to do. So just getting into the numbers game here, if you count them at the beginning, how many should you have in your cell? So this is before synthesis. So pre-synthesis here, how many sticks of DNA do you have? How many chromatids?
46.
You have 46, okay? That's your full complement. All right, so that number is called the diploid number. You have 46, that's your diploid number, or your 2n number. It's often called 2n. Once you make a copy of each of them, how many sticks of DNA do you have then? You've made an identical copy of every single one. How many do you have then?
92.
92, okay? You have 92 chromatids, but you still say you have 46 chromosomes, okay? This is a terminology game. Because these are identical copies, you call that one chromosome still, even though there are two sticks, okay? So this is still, everything in blue here would be called one chromosome, even though there are two chromatids in it. Okay, so 92 chromatids, but still 46 chromosomes. This stuff when I was in high school really irritated me, that there are these like little minor differences in the terminology, but anyway, you got 92. This should, by the way, this should say if you count individual chromatids, not chromosomes. Of course, there's an error in the actual note. Fantastic. Blech. All right, so I'm going to go through all the phases over again of mitosis, and hopefully it's a review for you. And then in the second half of today, or I don't know, second half of the second half, we're going to look at meiosis and how this is similar and different to meiosis. Meiosis is quite a bit more complicated though, so it's really good that you guys have this as background already. So again, interphase has three parts. You got the G1 gap phase, which is growth. They start off pretty small after they split. Then you got the synthesis part, which is making a copy of all of your DNA. Now you have sisters, sister chromatids. And then G2 is mostly centriole reproduction, although there's a couple other growth things that happen during that, but you don't really need to know what they are. Under the microscope, there's really not that much to see. While the DNA is unwrapped like this and is in its chromatin form, it's not really visible, so it just looks like a blob. Normally we would do a lab with this, where we would look at some cells in mitosis, but we're not supposed to use the microscope, so we can look at some pictures. It's not the most interesting lab you've ever done probably either, but it is kind of cool that you can actually see these under the microscope. But it is possible to see a difference from the beginning to the end of interphase in that the nucleus actually gets darker. And the reason it gets darker is just there's double the amount of DNA in there because it's replicating. So keeping in mind here that there are 46 chromosomes in both of these cells that are shown, but in the second one, there's 92 chromatids. You would still say that both of these cells have the 2n number of chromatids or chromosomes. They have the full genetic complement that a cell should have. Although in the second one, it's actually kind of like 2 times 2n. It's each one is doubled. And you would say that both of these are diploid. Okay, first phase of mitosis. Does anybody remember what it is? Without looking at your note? Nobody remembers. Anyone remember the little acronym from grade 10?
PMAT.
PMAT. Exactly. So the P in PMAT is prophase. So in prophase, you're going to take that chromatin, which is just DNA spaghetti, just laying out in a big bowl, and you're going to wrap those DNA noodles around some little proteins. Called histones. You don't really need to know what they're called till grade 12, but they're called histones. And then it will form those big sausages, chromatids. The centrioles will start building their little ropes, their spindle fibers, which eventually you're going to use to tug stuff around inside the cell to the right spot. I mentioned on here that the cytoskeleton breaks down. I don't know, you don't really need to know that, but there is a structure inside the cell that helps it keep its shape. It's basically a whole bunch of scaffolding that's built inside the cell. Did you guys talk about this in grade 10? Some people do in grade 10 and some people don't. Yeah, maybe you did. Cytoskeleton is like a bunch of proteins inside that holds the cell shape, but that all breaks apart during this process. It gets in the way, basically. If you're trying to tug big pieces of DNA all over the cell, you can't have scaffolding going all over. It'll get caught on it. So the cytoskeleton gets broken down. You start building your DNA tug ropes, spindle fibers. And those centrioles start to migrate to the opposite ends of the cell. Here's another pain in the butt terminology thing here. A centrosome is 2 centrioles put together. They come in pairs. So 2 centrioles is a centrosome. Each log is called a centriole, and 2 logs put together is a centrosome. That's sort of like the difference between Golgi body and Golgi apparatus. Lots of words for your vocabulary list, at least. I'm not sure if I warned people at the beginning of the course. This course has like 500 new words in it, minimum. Sorry, that's kind of like the downfall of biology, is it's so much vocabulary. Oh, and the nucleus falls apart. Yeah, I didn't mention that, did I? Nucleus dissolves. I put it on my diagram. That'll just get in the way. Got to get rid of that thing. By the way, that's why it's so important that you replicate the DNA before mitosis. Because once you dissolve the nucleus, now the DNA is just sitting out in the cytoplasm where it could potentially be reacted upon by all the enzymes and all kinds of crazy stuff in the cytoplasm. So you want everything to be done and like kind of connected together before you do that. So you don't want random binding sites just sticking out into the cytoplasm. You're gonna end up with all kinds of crazy things stuck to it. Can I keep going? Just make sure everybody's done. Okay. So the second one's metaphase. By the way, prophase, if you look at it, you're like, holy cow, lots of stuff happens there. It's by far the busiest phase. So it only gets easier from there. Not much happens in metaphase. So you get attachment of those spindle fibers, which tug the chromatids, two sisters now attached with a centromere, into the center of the cell. There's actually a physical structure there in the middle of the cell. It's a protein that's built across the middle of the cell called the metaphase plate. And it actually lines them up on that physical structure. You can't see it under the microscope because it's only a couple molecules thick, so it's not really much to see, but it is there though. There are minor differences in this, by the way, in the different kingdoms. Animals all do this exact thing. Plants are very similar to this, but with some minor differences because they also have to deal with the cell wall and a bunch of other stuff on the outside. So there's some extra things that happen, but you don't need to know them. But it's more or less the same for all eukaryotes. There's just minor differences. They don't have a metaphase plate, but they have something else in plants. So that centromere that's in the center of the X is actually right on top of the metaphase plate. Like the molecule binds to it. So it's actually what helps it break apart so that you make sure you get one stick going one way and the other stick going the other way. Okay, that's IP. What's the A?
Anaphase.
Anaphase. I like to call anaphase banana phase because it's when the X is turned into bananas. That helps me remember. But you use whatever you want to use to help you remember that. But all it is just the chromatids are being pulled apart. So the sisters break apart at the centromere and they get pulled to opposite poles. I mentioned in the little diagram here that this is where the cleavage furrow starts to form. That's like where the cell starts to pinch in on either side. That's actually cytokinesis, but it's starting right now before the end of anaphase. So cytokinesis kind of overlaps a bit here. And that furrow is built right on top of the metaphase plate. It actually is pulled on the metaphase plate. So that helps direct where this cleavage furrow is going to be. Make sure that you split that cell actually in half. You don't want it to be like 2/3 and 1/3, or you're going to end up with one funky cell. So far, this isn't like anything new for people, is it? This isn't like, oh my goodness, I've never. Okay, all right. I'm hoping this is review. It's tough because the end of last year was a real schmozzle. Who knows what happened in there? So. Should I keep going here? Okay, you've separated the DNA out to either side. Now you got to put it in a little protective bubble. That's telophase. So each new DNA package is going to get its own little nuclear membrane to protect it. And right after that happens, the DNA starts unwinding from the histones and goes back to its chromatin form so that it can start, transcribing stuff again. Ready to get back into the action. While this mitosis phase is happening, no transcription or translation can happen, no protein synthesis. So this can't be an extended period for a cell or it would die. It can't make any of the things it needs for life during this, while this is happening. So it's got to be relatively brief. That being said, some cells don't have a nucleus. Red blood cells don't have a nucleus, so they have sort of some special mechanisms to deal with that, but most cells do. Just wait for. And then lastly, cytokinesis kind of started earlier during anaphase, but cytokinesis finishes anyway, and actually splits the cell into two separate membranes. So it actually forms a loop, a protein loop that goes around the metaphase plate, and there's a motor protein that will pull the loop in, and as it pulls the loop in, it pinches the cell until there's two cells. And in the average cell, it takes about 10 or 20 hours, somewhere in that range, depending on the type of cell. So some cells are bigger than others, and there's other factors involved there in terms of the timing, but it's about 10 to 20 hours. Bacteria, because their process is so much simpler, can do it way faster than this. So they can do binary fission, and I don't know, some of them can do it in just 20 minutes. So they replicate rather quickly.
How long would the life of one of these cells be if it's 10 to 20 hours for that?
It depends on the cell type a lot. So if it's a neuron, like a central neuron, like in your brain, it could be around for 50 years. If it's an epithelial cell, skin, lining of your intestines or something like that, might only live for a couple days. So it really depends. Mm-hmm. That's why you may or may not have talked about this in grade 10, but if you have chemo, if you're getting chemo, it attacks rapidly dividing cells. It's trying to that the cancer cells are rapidly dividing, but so are your lining of your intestines, your bone marrow, your hair follicle cells. Those are all rapidly dividing, and so they also tend to take a lot of damage from the chemo, which is why you lose your hair and you have digestive problems and all of those things, skin rashes. Less now. Chemo is actually a lot better than it was 10, 15 years ago. It's much more targeted. Anyway, one quick thing to point out here is if you look at this cell, I color-coded them for your convenience. What is the diploid number of this cell? What's the full complement of chromosomes? How many?
Like the ones in there?
Yeah. What's the diploid number for this organism, whatever we just showed here? How many chromosomes does it have?
One? Isn't it?
How many are there?
Oh, 4.
4. This is the diploid number is 4, right?
Oh, okay.
If that was one of your cells, if that was a human cell, how many would be in there?
23. Oh,
This is
5
di-
plates, so 46.
46.
Yeah.
So that's key. I've only shown 4, okay? But if this was a human, there would be 46 here at the end. All right. Whenever I show examples of this, I'm only ever going to show you 4 or something, whatever I can fit like in a little drawing. But keep in mind that there are 46 for you, okay? Or 47. I mean, it's possible for you to have an extra one. That does happen. But typically 46. So all familiar? Question?
What would happen if you had extra chromosomes? Like if you had 47?
We're going to talk about a couple common cases right after we go through meiosis, and we're actually going to talk about how you get the extra one as well. So the process is called non-disjunction. So it depends on which ones you have extras of. Some of them have almost no effect, and some of them have very profound effects. So we're going to get to that in a little bit. One other thing to quickly mention here is that this process is not perfect. So in anaphase, ideally, you want them to separate at the centromere and pull one copy to either side. But you can get an unbalanced pull. So you can have it so that, well, maybe this one doesn't break apart and both go in one direction. If that happens, it usually activates apoptosis. So apoptosis is the cell's insurance policy. If you have too much or too little DNA, it usually just does cell suicide. So it packs up, instead of doing it in a unorganized way, that would be necrosis. Necrosis is basically when the cell just explodes. So you don't want that. That's when your cell is just has no choice but to suddenly die. Like if you get frostbite or something like that, that causes necrosis. But what you want is apoptosis, which is where the cell will just pack up all of its goodies, you know? Oh, here are all my proteins, let's put them in a little package. Here's my sugars, a little package. And then it'll lyse itself. So it does cell suicide, but it packages everything up nicely before it does apoptosis, so it doesn't trigger an immune response in your body. So apoptosis is the ideal way for cells to go if they're malfunctioning, and you want that to happen. If your apoptotic mechanism does not work properly, you end up with things like cancer. So you've got a cell doing something funny. It should just shut down and pack up shop, but it just keeps going. And you don't want that to happen. So apoptosis is very necessary. It sounds macabre, I guess, for cells to commit suicide, but you don't want rogue cells doing all kinds of stuff. Okay, I'm going to leave these this part at the bottom here for you to do actually at the end. Okay, so maybe in learning block 3. These are just some standard sort of review questions from mitosis. Okay, so I'll get you to go back and do this in a minute. I did want to mention this part at the bottom here really quickly. This is sort of a general thing that you talked about, hopefully in grade 10. Do you guys talk about why cells divide in the first place? Like, why don't they just get really big? Like, why aren't you like one big cell? It's a valid question, because if your cells can keep growing and growing and growing and stuff, like, why don't you just have really big cells instead of, 35 trillion or whatever you actually have? A lot, something like that. So there's a really good reason for it. And the reason for it is that you need to get all of the stuff in your cell, oxygen, sugars, whatever, to support what's in the middle. It all has to come across the cell membrane. And you can only transport so much across a cell membrane at a time. So there's like a maximum amount that you can pass across a cell membrane. Get oxygen in, get sugar in, get waste products out, get carbon dioxide out. There's a maximum flow rate, okay? So what we're really talking about here is surface area. That's the outside of the cell, right? You need a certain amount of surface area for the amount of stuff inside that needs the resources. All the different machinery that's using oxygen and producing waste. And so the amount of surface area you have to the volume inside, that's your surface area to volume ratio. So as a really quick demonstration here, and I have a feeling you guys are familiar with cubes from math. We could do this with spheres too, but the math is easier with cubes, and it ends up being the same, more or less, okay? So if you have a 1 by 1 by 1 cube, okay, you can calculate the surface area of that, right? It's got 6 sides, they're 1 by 1 each. That's 6 units cubed, or 6 units squared. That's the surface area around the outside, right? And you can also take the volume of that cube. That'd be 1 by 1 by 1, 1 cubed, right? So that's 6 units of surface area to 1 unit of volume. Or you could also say that as a 6-to-1 surface area to volume ratio. That's how much capacity you have to supply the inside versus how much inside you have. Okay? If I double the size of this cube and I make it a 2 by 2 by 2 cube, then the surface area of that is going to be 2 squared, right? That's one side, times 6. So that's going to be 24 units squared, right? That's the surface area, 24 units squared. And then the volume would be 2 cubed, 2 by 2 by 2. So that's 24 to 8, okay? And if you simplify that, that's a 3-to-1 surface area to volume ratio, okay? You'll notice that it's less surface area to the amount of stuff inside of the cell, okay? In other words, the stuff inside grew faster than the stuff outside that keeps it alive, okay? If I make it 1 unit larger and make it a 3 cube, now we've got 54 as the surface area and 27 as the volume, 3 cubed. So now the surface area to volume ratio is 54 to 27, which is 2-to-1. So the bigger the cube gets here, the more volume there is to the surface area that supplies it. In other words, there's less stuff on the outside to support the stuff that's on the inside. And eventually this gets to a ratio that becomes impossible to support the stuff on the inside. There is a maximum size of cell. They can only get so big until you can't support the stuff inside anymore, and you have to make more cells. You need the surface area. That is the reason why cells, in general, in nature, are more or less the same size in terms of orders of magnitude. Okay, so if you look at your cells and the cells of a monkey and like the cells of again, you can go outside the animal kingdom, the cells of a mushroom or something like that, they're relatively similar in size because the surface area to volume restrictions are the same on pretty much all organisms. Bacteria typically have smaller cells, but that has more to do with their complexity inside than it does with the surface area to volume problem. But they're relatively similar. One of the other cool things about this is that the reason that there is difficulty getting stuff inside also has to do with concentration differences. So the higher the concentration of whatever gas on the outside, let's say, for example, there's more oxygen in the air. Let's say it was 50% oxygen in the air instead of 20. That would mean that there's more pressure to push oxygen into the cell. And so you can actually support more volume with less surface area if you have a higher percentage of oxygen in the atmosphere. And that's why if you go back in the fossil record, you can find insects that are like, the size of a car. Like there's just like giant insects that exist because organisms could have larger cells. Things could be larger, and this is like allowed within that system, which is kind of cool. So get giant insects. Not going to find those anymore. The atmosphere won't support that. But it's really cool to think at one time it did. There was more oxygen in the atmosphere at one point. Anyway, I just mentioned this right here, the idea of this. You don't need to worry about this too much for this course, but that's really why we're talking about mitosis, why mitosis exists, because you need to make new cells to respect this boundary. Okay? Do you guys know that I post these notes? Everybody knows this? Okay, so it's in the classroom notes folder. If you ever need to go back and look at any of this stuff, it's always available to you in there. Okay, the completed versions are in there. Anyway, these other questions I'm going to leave for you to work on after. What we're going to do right now is change gears a little bit and talk about, instead of making adult cells, somatic cells, which is what mitosis is all about, we're going to look at how to make gametes. So sperm cells and egg cells, because obviously it's got to be a different process. If you ended up at the end of that process and you produced a 2N cell that had all of your chromosomes in it, and then your partner, whoever you were going to make a baby with, also did that, well then you would end up with way too many chromosomes at the end, right? You'd have 92 chromosomes. That's fatal. So cells can't survive with 92 different chromosomes inside. So there's got to be a system here. We've got to pare it back. You're only allowed to give half of your genetic complement to your children. So it's random though, which is probably for the best. You get to pick. So if you're looking for the note for the second half here, There is, by the way, you don't have to watch the second video on mitosis. I would say if you didn't cover it last year in grade 10, this is another good summary video. And it also talks a little bit about surface area to volume ratio, which we just discussed together. But again, if you did it last year, I wouldn't worry about it too much. But the second part of the note is right there, the beginning of meiosis. I'm going to give you guys a heads up. This process is the number one thing that people find confusing in this course, okay? It is complicated and long and a little bit confusing. And if you don't understand immediately what is happening once we go through it together, I would say that's normal, okay? So give this one a little bit of time. We're gonna get through about half of it right now before the end of the class. I highly recommend this video, which is gonna go through the basics of it again, which I would watch in the interim, okay? Consider watching this during learning block 3. You don't have to, but I do highly recommend it. But I will say that this particular process is the thing that people have difficulty with in this course. Just like I think for a lot of people, mitosis was something that people had difficulty with in grade 10. But this even more so. This is quite complicated. So don't feel bad if it's a little bit confusing at the beginning, and we'll get through it together. We're going to do a project on this. We're going to do a little review in the classroom with some pipe cleaners, and we'll do some other stuff. Okay, so don't be too hard on yourself. This part is a little bit complicated. So do you guys all have the note out? Yeah, you guys are fast. Where are you here? Genetics. All right. Meiosis. The top of your note looks just a touch different than this, but it's the same note, I promise. A good question to ask. Why sexually reproduce? It's such a pain. You need to find a partner, which is a pain. You need to go to all this trouble of producing sperm or eggs. In the case of sperm, you're making, billions. You're only using one of them. It's a pain. It's a complicated process. In nature, not every organism uses sexual reproduction. There's lots of small organisms that just bud or produce an identical copy of themselves. And there are even larger organisms like fish and reptiles that clone themselves, that literally produce a fertilized egg inside of themselves that is a clone of the parent. So it's an option. You can do clonal reproduction even with large multicellular organisms. But in general, it's almost never done. 99.99% of the living world is sexually reproducing. And I'm kind of excluding a lot of the species of bacteria here. Bacteria are not capable of sexual reproduction, as you're probably already aware, because they're just making copies of themselves, binary fission. But they do something similar where they, I can't remember if I talked about this in the note, where they create a little tube and they give some DNA to their friend next door, Apillis. Did I talk about that? Yeah, so they have that, but it's not really sexual reproduction because it's not like a half and half. It's just like hey, what do you think about this gene? Take one of these. But that's pretty much it. So the thing I want you to get out of this, the reason why this whole process exists that we're going to talk about is because of diversity. And this goes back to the first unit, the very first day, where we talked about how diversity is strength for a species. It's survival. The more diverse your population is genetically, the more likely your whole population is going to survive any particular adverse event. Change in temperature, change in location, change in food source, whatever. Whatever it is, if you have more diversity in your population, you're more likely to survive. And sexual reproduction creates diversity. A whole bunch of the different parts of this process that we're going to talk about right now are there for the only purpose. They're very complicated, but they're only there because they create diversity. So that's the important part of this, okay? We're going to really harp on this because the importance of diversity is kind of one of the main things that comes up in every single unit of the course. We talk about it in evolution, and the diversity unit, in this unit, pretty much every unit. So this is that's the really important part here, okay? So a little bit of terminology there at the top. I did replace one of these. I I can't remember if I replaced the one that's in your note. I was looking this over, and non-sister chromatid is a very confusing term. I'm going to replace it with non-identical sister chromatid. We were just talking about sisters, right? This idea that you've got two identical ones that are attached in the middle with a centromere. In this process, we're going to rejig them a little bit so that they're not identical to each other anymore. And I'm going to call those non-identical sisters. But we'll get to that in a bit. Okay, so what is meiosis? I've already mentioned this here. It exists so that sexually reproducing animals, not just animals, plants, any eukaryote, can produce gametes. So gametes are sex cells. Cells that are there for reproduction. Often they're called sperm and egg. In the animal kingdom, they're always called sperm and egg. In the plant kingdom, sometimes they're called pollen and egg. In the fungal kingdom, they're called plus and minus. I think I talked about that in diversity, the plus and minus thing, and they've got a real funky thing going on there. But it's the same thing. So they do meiosis and produce their plus and minus. So. And what's going on here is you're taking your diploid cell, a 46 chromosome cell. We're going to name them in a second, but they're called boy, I got it later in the note. I'll come back to it. Spermatocyte and oocyte. There we go. And those are going to be used to produce a sex cell, a gamete. Our diploid number is 46, but obviously it's different depending on the species. So if you're a chimp, it's 48. If you are a fruit fly, it's 12 or 8. Something like that. Anyway, it's different depending on what kind of organism you are. And obviously, if we took your full genetic complement, your 2N cell, and just smashed it into somebody else's 2N cell, we'd end up with a condition called polyploidy. Polyploidy is when you have too many of a specific chromosome. It doesn't have to be a full doubling. Well, that would be the absolute worst case scenario, but even if you had too many of chromosome 1, like 3 chromosome 1s, you'd also call that polyploidy. So polyploidy is too many. Most of the time, polyploidy is fatal. So if you have 3 chromosome 3s or 3 chromosome 10s, it's usually fatal for most organisms. Humans have a couple chromosomes where they could take it. You can have a viable offspring where there's 3. Most of them are fatal though. In plants are surprisingly resilient when it comes to polyploidy. So some plants actually can easily accept extras and it doesn't really affect them at all. So we'll leave that for a different time. But some organisms are more accepting of polyploidy than others. Okay, so what you need to do then in order to get all this to work is you got to fuse those specialized sex cells together, or gametes. There's 2 separate processes here, but they look identical more or less. We're going to talk about the differences between the male and the female process, but it's very similar. There's a couple small deviations. In males, that process is called spermatogenesis. So spermatogenesis is meiosis. That's the same thing, but it's just the male version of meiosis, spermatogenesis. And in females, it's called oogenesis. Some people just say oo, and some people say oo-oo. So I'll leave it up to you. You can call it oogenesis or oogenesis. I had profs in university that did it both ways. So in men, you start off with what's called a spermatocyte. That's a 2N cell and then you end up with a sperm, or it's sometimes called spermatozoa, but that's the same thing, sperm or spermatozoa. That's the 1N version, when they've given up half of their genetic complement. And that happens in the seminiferous tubules of the testicles, for male humans anyway. A lot of, certainly all mammals, many animals have similar structures. For females, the cell that you start off with is called an oocyte. And then once you go through meiosis, you end up with an ootid at the end. An ootid is an egg. That's an O, by the way. It's OOTID. Ootid. And that happens in the ovaries. It's important to note that the spermatocyte, so that's the cell before you do meiosis in males, is also doing mitosis because you have to make more spermatocytes all the time. So it actually does both. So it'll do mitosis very regularly to make more spermatocytes, and then some of those spermatocytes will go on and do meiosis and produce sperm. So you gotta do both. For females, you've already done all of your oocyte mitosis. So you have all your oocytes that you're ever gonna have. That actually happened in the womb. So you've got all your oocytes, and now they've actually started meiosis. They've gone through part of meiosis, and then it's actually locked partway through meiosis, and then you get to pick an egg or two, an oocyte or two, to continue and finish meiosis every cycle. But all your oocytes are already present. They've done all their mitosis, so it's a little bit different. So again, some important terminology here. The sex cells are haploid, which means they have half of the total complement of DNA, also known as N, or 1N. So if you take a sperm, 1N, and an egg, or an ootid, same thing, and combine them together, you end up with a zygote that is 2N, or has 46, or you can also call it diploid. The nuclei actually fuse together, combine the two. It's weird to think that, I don't know, maybe it's not weird. It strikes me as weird that when you make a zygote that will eventually become a baby, you are physically taking a cell from the mom and the dad and fusing them together into a single organism. It's kind of cool. You are a fusion of your parents. You're actually a cell from your mom and a cell from your dad that's been fused together into a single organism. All right, let's go through the process of actually making these things. You're going to notice that, hey, this looks just like mitosis. Yep, it looks just like mitosis, except it's twice as long and twice as complicated. But it is a lot like mitosis. It's broken into two parts. The first part is called reduction division. It's like the first half of meiosis. It's called reduction division. And the purpose of it is to take 92 chromatids and separate them down into two 2N cells. The sisters, that is the identical pairs, are going to stay together, and the homologs are going to get separated. In other words, your maternal DNA is going to go one way and your paternal DNA is going to go the other way, but you're going to keep the sisters together. The things that are stuck together with the centromere in the middle, those stay together during meiosis 1. So sisters stay together, but homologs separate. Yeah, if that doesn't make any sense to you, well, we'll get there. Go through the whole process here and it'll become more apparent. Also note that, again, it is the same names of the phases: interphase, prophase, metaphase, anaphase, telophase, cytokinesis. You'll see them all, but they have a little 1 after them or a little 2 after them because you go through it twice. So there's no such thing as interphase of meiosis. There's only interphase 1 of meiosis and interphase 2 of meiosis. If you ever see it written and there's no 1 or 2, you're talking about mitosis, okay? Mitosis doesn't have any numbers. But if you're talking about meiosis, you got to put a 1 or a 2. Otherwise, who knows what you're talking about, because they're different. All right. We've started off here with an oocyte or a spermatocyte. It's identical at this point, so I'll let you know if they ever deviate, the male and the female version, but right now they're identical. You start off with a big old cell, and you have to do the exact same thing that you always have to do during interphase, which is what? What has to happen during interphase? We did review it, but I don't know if anybody remembers. That's okay if you don't. What happens during interphase? That's prophase. Okay, growth? Yep, that's one. Oh, sorry, what was the second part that you said? Synthesis. Okay, so you got it. So growth and synthesis. So you're going to, the cell's going to grow. I didn't include the growth part here because it's not terribly important. But the part that we really care about is the synthesis part. You're going to make a copy of your entire genome during interphase. So just like mitosis, you're going to make an exact copy of everything. So your cell goes from being a 2N cell to 2 times 2N, just like it does in mitosis, okay? In fact, at this point, this is no different from mitosis at all. It actually looks exactly the same. I wouldn't call this 4N, because 4N implies that the other set of 2N is different from the first set of 2N, and they're not. They're identical to each other, right? So this is really 2 times 2N. It's one version, one 2N, but there's 2 of them. So far we're good? Okay, cool. That one's not the most complicated step. All right. This is when it starts getting crazy. So prophase, as always, is like your crazy Uncle Zeke or whatever. It's the crazy step that has a whole bunch of stuff happening. So you've made a copy of everything. What happens in prophase of mitosis? Let's start there, because there's very clear crossover here.
The nuclear membrane dissolves.
Okay, nuclear membrane dissolves, exactly the same. So that happens here. What else?
The chromatin condenses into chromatids.
Exactly the same happens here. Okay, chromatin condenses into chromatids. This is when things start to get a little bit different. Okay, now what's going to happen, and you can see the nucleus dissolving there, by the way. Nucleus dissolving, chromatids are formed. You can see the chromatids. But now the homologs, okay? So remember, these are not identical to each other. They are the mom version and the dad version of each chromosome. The homologs are going to give each other a hug. That hug is called synapsis, okay? It really is a hug. They just get together and wrap around each other. Once they've hugged together, now there's 4 chromatids stuck together in a hug, in a synapsis. That's called a tetrad. Okay, that's what the name of the structure once they've all gotten together. Okay, it's shown in this diagram as just having a partial overlap. That's just so you can see what's going on, but they really do intertwine with each other. I also mentioned on here that the nuclear envelope dissolves. That stuff is exactly the same as mitosis. Now, obviously in mitosis, you don't get the homologs hugging each other. There's nothing going on with the homologs whatsoever in mitosis. But in meiosis, they hug. Okay, everybody with me so far? Once they hug, they actually exchange a little bit of their DNA with the other homolog. They actually say, hey, what's your gene for hair color like? Here, have mine. And then they'll just switch it up. Oh, okay, I'll take your hair color. Would you like my gene for toenail thickness? Done. Here, you can have this toenail thickness, I'll take the other one. And you get this exchange happening. So that exchange between the two is called crossing over. Oops, it isn't. That's this right here. So crossing over is an exchange of DNA between the homologs. And because they have the exact same genes on them, it doesn't matter. You can switch that genetic information as long as it's coding for the same thing. So blue eye color, oh, take my brown eye color. Here, you take blue. And then they just switch with each other. They only switch at specific random spots, which means every single time you do this process, what they switch is different. It's completely randomized every single time. And that's important because it means that the gametes that you get are completely different every single time you do this. You could have literally 1 billion babies and you would not get genetically identical babies. If you just did it over and over and over again, you never get genetically identical babies because you make your gametes different every single time. Question? Twins are a special exception. So twins are actually had one zygote. You're talking about identical twins, right? Monozygotic. So that's when you took a sperm and an egg and made a zygote. So now you got your 2N, right, in the zygote. And then that zygote actually splits early on. Sometimes it's not always the first division. Sometimes it's like 100 divisions in. But it actually splits into 2 separate organisms, 2 different fertilized organisms. So, and then you end up with two babies. Now those are genetically identical, but that's not from this process, right? This cannot produce 2 identical, but it is possible to get identical. So what I'm showing over here is the actual synapsis occurring. So let me see if I can draw this again here. So you've got your chromosome 1 from dad and chromosome 1 from mom. They come together and do a little hug, a tetrad. They form a tetrad. This is called synapsis. And then they're actually going to switch like a little piece of their leg. You know, like this guy's going to be like, hey, take this little piece. Cool, here, have this one. And they do a little switch. Okay, so that's the crossing over. That's a huge exaggeration. They don't switch like their entire leg or whatever, but it's like little pieces. So that's kind of hard to show on here. That's what's being indicated with the letters there. Those letters are versions of a gene. They're called alleles. We're going to get into that more in the second half of genetics. You don't need to worry about the letter stuff quite yet, but we will get into that. But as I mentioned earlier, the purpose of this is increase in genetic variation. You want that. That's our strength. That's how we survive as a species. So overall, this process, all of the different chromosomes doing this, is called genetic recombination. I just have that written at the bottom here. So genetic recombination is crossing over, but like on a large scale. It's when you do all your crossing overs. That's called genetic recombination. And because you did that, if you look at the chromatids that you end up with at the end, if you actually look at these chromatids, you'll notice that they're all different. Come on. So if you look at all 4 of those up there, none of them are identical to each other anymore. So we've created a whole new set. They're still stuck together in the center. These sisters are still together, but they're not identical to each other anymore. So this is what I'm calling non-identical sisters. They are sisters, chromatids, but now they've done a little switcheroo, so they're not identical anymore. Are you guys good with this? Yeah? Okay. Let me know if I'm going too fast. I actually usually have the opposite problem where I talk too much and I go way too fast, but I'm trying to slow it down. So, okay, that's prophase. You will be very happy to learn that that's the longest and hardest part, okay? Just like it is in mitosis. So way less happens now. What happens in the next phase of mitosis, in metaphase? What's metaphase all about? What do you do? Line them up in the middle. Okay, so metaphase 1 is exactly the same. You get your kinetochore, that's the binding sites that are on the centromeres, and they're going to get attached to the spindle fibers, and the spindle fibers are going to shuffle those chromosomes, the chromatids in this case, chromatids, into the center of the cell to the metaphase plate, with a minor difference. Normally, these would be arranged on the metaphase plate, right? They'd all get lined up on the metaphase plate, but now they're going to be arranged so that each pair, that is chromosome 1 from mom and chromosome 1 from dad, are on opposite sides of the metaphase plate because they're still in their tetrads. So this one and this one are, I didn't do a great job there, are still stuck to each other in a tetrad. The tetrads are still there, but they're going to be lined up so that one of the pair of sisters is on one side and the other pair of sisters is on the other side. In other words, homologs are on opposite sides. One really important thing to note about this is that which side the paternal goes on and which side the maternal goes on is random. So it could be arranged like this, or it could be arranged like this. Which way it's arranged, that the paternal or maternal's on the left or the right, is random. And remember that there's 46 or there's 23 of these pairs, right? And so every single one is random. So it could go paternal, maternal, maternal, paternal, 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 maternal, totally random every single time. So the first chromosome could be like this, the second one could be like this, then the third one's like this, and the fourth one's like this. Totally random, okay? So that randomness of where you're going to get the maternal or paternal to go is called independent assortment. And independent assortment is also important. Because it's random, it again means that there's going to be more variability in the offspring. Anytime you add an element of randomness to this, it means more differences in the potential sperm or eggs that you're going to produce, which is good for diversity. Diversity good. That would be another great thing to add to your vocabulary list. Independent assortment happens during metaphase 1. I already mentioned this, but yeah, not on the plate. They're on either side of the plate. Cool. You guys get? Yeah. Okay, so that's metaphase 1. Anaphase is next. What happens in anaphase? In mitosis. Yeah, it's going to be an approximation here. Julia? Exactly. And it's exactly the same thing here. So in anaphase 1, those microtubules, or spindle fibers, same thing. Spindle fiber is just a type of microtubule, is going to pull those homologs to either side. And it's really important to note here that unlike mitosis, the sisters are not getting pulled apart. So the sisters are still stuck together with the centromere in the middle. But the homologs are getting pulled apart. So now they're not in tetrads anymore. They're getting yanked out of their hug, basically. Okay? So it's like, no more hug for you. They get pulled apart. And paternal will go one way and maternal the other, and it's random which way they go. So when you make a sperm or an egg, and it's going to be left or right here, it's going to be a mix between paternal and maternal genes. So in other words, when you make your own sperm or egg, it's not going to be all of your father's genes or all of your mother's genes. It'll be a mix of your father's and your mother's genes that will go into each one. It's random. I mean, I guess technically it is possible for it to be all fathers or mothers, but it would be very rare, a total random coincidence. If you've ever wondered why, sometimes two siblings, maybe you and your brother or sister, look very similar, or sometimes they look very different, this is a random process. So yeah, sometimes you get similar splits that go the same way, and then wow, that really does look similar. And sometimes you get completely different splits, and you look completely different from each other. So anything's possible. They're still going to have your genes either way. So it's not like they're not getting your genetics, but which combination of your genetics is the question. Okay, and then lastly, telophase, the end of mitosis. What happens in telophase?
The chromosomes decondense at opposite sides.
Right, you got to put them in a nuclear envelope or in a nucleus. Okay, telophase 1 is exactly the same. You got to put the DNA in a nucleus. I didn't show the decondensing happening here, but it is happening. There's a decondensation here happening as well. Do you want me to turn the heat up?
No, they were bullying me before because I put my jacket on twice today.
He puts it on.
Keep
and
taking
he's
it off
taking
and
it on
turning it on
and
again.
It's thermoregulating, guys. It's normal.
Bullying, guys.
He's having to walk backwards. Yeah, walk back.
What are you talking about?
And we're not flush.
And then lastly, at the end of meiosis 1, you got cytokinesis 1. I forgot to put the 1 in. There is supposed to be a 1 there. It's supposed to be cytokinesis 1 because there are 2 of them. So now you've got your DNA all decondensed and you split them in half. Now now we're in a sort of like a weird gray area here because each of these cells does not have 2N. 2N is the full genetic complement, right? But you had one, like maternal or paternal, go one way or the other. So you don't actually have a full 2N in each of these cells anymore. You have a little less than 2N. So I like to call this 2 times 1N. You have 1N and an almost identical copy of that 1N, but it's not quite 2N. So it's a little less than 2N and a little more than 1N. I'm going to call it 2 times 1N. All right, a little bit complicated here. But you definitely have way more than 1N. So you can't use these. These cells are no good to make a baby. If you put one of these together with another one, you'd have way more than 2N at the end, and then you'd end up having polyploidy, too many chromatids. So we're not done. We're not done. But we do have two non-identical cells at this point, totally unlike mitosis, right? In mitosis, these would be the exact same cell. But here, their genetics are completely different from each other, which is what we want, diversity. That's why meiosis 1 is called reduction division, okay? Because there are two approximately haploid cells now. We've reduced the genetic complement in each of these cells. Each one of them has two haploid sets in it and not a diploid set. In other words, what we did during meiosis 1 is separated the homologs. The homologous pairs are now separated from each other. So there's not two copies of each chromosome. There's not two chromosome 1s and two chromosome 2s and two chromosome 3s. We've separated them. If you just stopped right here, like I said, you wouldn't be able to use them as gametes. You also wouldn't be able to use this as a regular cell because there's not enough genetic information in either of these cells to operate properly either. So, like I said, this is like a weird gray area where the cell's couldn't be a regular somatic cell and it couldn't be a gamete. So if it stopped at this point, apoptosis is the only option. Can't use these for anything. But we can fix that. We good? Yeah. All right. We can fix that with meiosis 2. So meiosis 2 is all about separating sisters. Sisters are still together. We got to split them up. And so meiosis 2 actually looks pretty much exactly like mitosis. It's very, very similar. And we don't do all the crazy cuckoo banana stuff of prophase 1 with the crossing over and the tetrads and whatnot. All that stuff is done. So this one is a lot more straightforward. One of the major differences, though, is that in interphase 2, normally interphase is all about DNA replication, right? But we do not want to make another copy of the DNA. If we made another copy, we wouldn't be able to get it down to 1N at the end because we would have way too much DNA. So there is no DNA replication that happens in interphase 2. It's just growth. So there's a little bit of growth and remaking of centrioles. You need the centrioles for organizational purposes, but there is no synthesis phase. Okay, so no synthesis. Then in prophase, this is going to look just like mitosis. Prophase 2 is the nuclear membrane breaking down, spindle fibers forming, centrioles migrating to either end of the cell. Again, it looks exactly like mitosis. In fact, if you looked at this under the microscope, this is indistinguishable from mitosis. You can't actually tell a difference. For my diagrams here, they got a little chopped up. Okay, what happens in metaphase of mitosis?
The chromatids get lined up along the center.
Line them up, okay, on the metaphase plate. Now, just like mitosis, this time the sisters are going to be on either side of the metaphase plate. So again, this is indistinguishable from mitosis. It looks exactly the same. And then anaphase 2 looks exactly like mitosis again. The chromatids get pulled apart. X's turn into bananas. And they go to opposite sides of the cell. And then in telophase 2, again, exactly like mitosis, you put each set of that DNA into a nucleus and they start to decondense again. And then the last part here, cytokinesis 2. I forgot to put the 2 on that one too, but cytokinesis 2, you have another separation that happens on the metaphase plate, and they get split into 2 cells. If this is happening in a male, what you get at the end, because there is a difference between males and females, you get 4 sperm. Each one is the same size, and they are just 4 different sperm with 4 different packages of DNA in them. They are not identical to each other. Each one will make a different offspring, potentially. Although probably they won't, but they have the option of. If it's a female, you only actually get 1 egg out of this deal. So what ends up happening is you get an uneven split. Every time the cell splits, it's not the same size on either side. So let me show you what I mean here. So if this was a female egg, the first time you get your split, so this is in cytokinesis 1, you're going to end up with a little one and a big one, okay? And then when they split the second time in cytokinesis 2, the little one just splits into 2 little tiny ones that are basically useless. And then this one splits into a little one and a big one. Okay, it's an uneven split each time. And then at the end, this is the only one. This one is the only one that is viable. That one becomes the ootid, and the other 3 are called polar bodies. They just get reabsorbed. So they basically nothing happens with them. They apoptose. The reason you have to do that as a female is because you are providing all of the machinery for the first 1,000 divisions, once the egg is fertilized, on board. You have to have many hundreds of mitochondria. You have to have hundreds of Golgi. You have to have hundreds of everything, because you are providing all of the resources for the first divisions of life before that fertilized egg, that zygote, can implant and start gaining resources from the mother. So before that happens, there are no extra resources for you. If you're a chicken, you got to put the whole thing into your egg because there is no connection to the mother. So that egg that's coming out unfertilized is 1 cell. It's hard to imagine, really. It's 1 giant cell. It's got a bunch of nutrients in there and cellular machinery, plus a nucleus. And then there's going to be a whole bunch of cell divisions that occur inside there, which is crazy. But humans have it a little bit easier because they get a little bit of support there from the mother. So it's more complicated at the end. But you still have to do all of the initial divisions based on what's in the egg. So that's why you had to have this unequal division. For the guys, for males, it's basically just a DNA rocket. It's got DNA in the front, a propulsion mechanism at the rear, and that's it. That's it. There's nothing else in there, guys. Maybe 1 or 2 mitochondria because you got to keep the tail going. And then other than that, that's it. No Golgi. You don't have to put anything on board that thing. It's just a DNA missile, and then it's like fire and forget. You just like hope it gets to the egg, and if it doesn't matter. You didn't put a lot of resources into it, so you don't have to worry about it too much. Females have the exact opposite strategy, which is put all the resources into a very, very small number of eggs because you have to provide so much more resource. This ootid that you get at the end, interesting fact, is the only human cell that is visible to the naked eye. So in ootid, if you like put it on a piece of paper, you can see it. It's really great. There's no other human cell that you can see. There's nothing that's big enough to see with the naked eye. But an ootid is large enough to see. That's how big it is. So it's got to have a lot of stuff here. I'm going to write that on here because I think that is so cool. And it's obviously way bigger than a sperm cell as well. So the sperm is just like a tiny little DNA thing that it attaches to it. So I mentioned here at the bottom that the process of meiosis 2, the whole second half here, is just for separating sisters. And the reason that this process is so important is because it has 3 elements that allow it to create diversity in offspring. The crossing over, which we mentioned, okay, random exchange of genetics between maternal and paternal. Independent assortment, which is the direction that the chromatids go when they're separated. And one last part, which is the pairing. Which sperm makes it? Which egg makes it? It's random. So that randomness also adds to the potential randomness of the offspring. It's pretty wild that all these things have evolved. But I mean, nature does what works. Okay, what I want you guys to do now, I'm gonna stop right here, is watch this video. I think you have just enough time. Actually, maybe you don't. You don't. Okay, I'll leave it up to you. You got some questions that you can work on. The questions are in the, are these right here, the cell cycle questions. Okay, so you can work on these. But I highly recommend that video as well. The first couple units are really the most full of information, and I realize that it seems like there's like a bajillion things that we're doing in here. It calms down a little bit towards the end of the course, just so you know. Okay, so I really like action pack the first little bit here. It's probably better for you this way, that it peters out as opposed to doing this. So, but we gotta squeeze it all in. So anyway, give these questions a shot, and then I highly recommend the video to watch in Learning Block 3. Question?
I have a question about the rest of the year. So you know how you said there's like a project to test and a vocab thing for each unit?
Yes, that's right.
Are they all like graded the same, like they're all worth the same amount?
That's correct.
Okay.
Yep, so each unit is worth 20%, and they're just compiled all together at the end.
Okay, and is like the project worth more than the vocab? Or
It is, yep. The project is worth by far the most. It's worth more than the quiz as well. The quiz is open book. I can only make it worth so much,
Yeah.
but the project is basically the majority of each unit. Yep.
Okay.
Do you also have the
Okay, for you guys at home, if you have any questions, sorry, just 1 second. I will be in the Google Meet in Learning Block 3, and please come and find me there.